This session is called Making Sure Plant-Based Foods Are Safe. And we'll start with Senior Scientist Tina Bech Hansen from Research Group for Food Microbiology and Hygiene. And Tina, you're going to talk about food safety of plant-based foods, the role of Bacillus cereus. So thank you for that.
Thank you, Christine. Bacillus cereus. Why are we concerned about Bacillus cereus in plant-based foods? There's actually 3 reasons to be that. The very first one is that Bacillus cereus is a foodborne pathogen, and it can cause 2 kinds of syndromes. It can cause diarrheal syndrome, where by ingesting large numbers of cells, we can get diarrhea. We can also have an emetic syndrome from this organism, and that is something that arises because it has an ability to produce a toxin while it is growing in the food that can make us ill. So now the EFSA Biohazard panel has looked into the cause of outbreaks from Bacillus cereus and made a report of that, and their conclusion was that we need more than 100,000 Bacillus cereus per gram to cause an outbreak. So remember that number. I will get back to that. Yeah, of course. Can I continue talking? Yes. Okay. So that was the first reason. The second reason why Bacillus cereus could be a concern is that the reservoir of this bacteria is soil. So when a bacteria is present in the soil, it can contaminate the plants very easily, and it can also spread all over the environment because of, for example, the farming practices that we use that will spread it all over, but also it can be carried by animals, so it can spread to other plants and other places. The third thing that actually is important for this organism is that Bacillus cereus is what we call a spore former. It means that it has the ability to get into a stage where it is extremely resistant to harsh conditions. And it is, for example, very resistant to heat. It is also very resistant to dry environments, and it is resistant to UV light, for example, coming from the sun. So this makes Bacillus cereus to survive for very extreme long periods in the soil, adding to the contamination risk for this food, for these plant-based foods. So when we look at these 3 reasons together and try to see what is then actually the concern when we talk about Bacillus in plant-based foods, we can say that Bacillus cereus must be present in the food, and it must also be able to grow in the food to cause problems for us. So therefore, we for the past time has worked together with the Danish Veterinary and Food Administration and actually looking into this and looking at how often and how many Bacillus cereus do we find in plant-based foods, and can it grow there? Or can we create situations where it will stop growing to have some safe foods? So, and these would be the 2 topics of my talk today. And I will start looking at how often and how many we can expect. So now we have looked into the literature to see what to expect. And we have also looked into the Danish data, because the Danish Veterinary and Food Administration actually has a very large database where they collect all the results that they have analyzed years back, so we can extract data from there also. When we look to the literature, very recently there was published a systematic review. It looks at the studies conducted between 1990 and 2020, where they have estimated the prevalence of Bacillus cereus in all kinds of foods and also in plant-based foods. So here I have taken the plant-based foods to show you. So the first thing I would like you to notice is that in general for these plant-based foods, we should expect approximately 40% to be contaminated. The other thing I would like you to notice is that it appears from this study that products containing cereals and products containing beans will have even higher prevalences of this organism. So this is the first conclusion, that it is actually quite prevalent. But a study like this, talking about prevalence, do not tell us how many is present, because it's a study where they look at many different studies that use different detection methods, that have different detection limits. So actually we don't know anything about that. So therefore we also look into the Danish data, where we have some controlled experiments, where we know the method and we know the level of detection, so that we can get an idea how many to expect. So the most recent Danish studies we can see in the databases are from 2017, where we looked for Bacillus cereus in heat-treated foods, all kinds of foods, and also plant-based foods. And we looked immediately after heating to have an idea what is in the food from the beginning of the shelf life. The second study that I would like to talk about is from 2020, where we looked in ready-to-eat salads to see how often they were contaminated with Bacillus cereus and how many to expect to be there. Also here, we looked at the beginning of the shelf life of the products. So, and I would like to start with the salads to show you some of the results. So how often and how many do we expect? So if we start with the prevalence, so how often do we expect to find Bacillus cereus in a vegetable salads? And you look at the numbers, you can see that it is around 30%. So, and 30% is lower than 40% that we saw in the literature. But we think the reason is that we don't know in the literature data the age of the food. Because if Bacillus is present in a food where its growth is supported, we will have a higher prevalence the older the product gets. And here we are in the beginning of the shelf life. So it fits very well. Also, when we look at the trend of cereals and beans actually giving rise to higher prevalences when they are added. When we then go to the other end of the scale, how many can we find of these bacillus in the product, and look at how often do we find concentrations of more than 10,000 per gram, we see that it is the same trend, that cereals and beans will also have give rise to higher concentrations of bacillus in the foods. And I would like to draw your attention to this 1% of the vegetable salads with the cereals included, where we actually have exceeded the acceptable level that we saw from the EFSA biohazard panel. So, and that indicates that there could have been growth of bacillus in this product. And this growth, as this is in the beginning of the shelf life, this growth has probably happened before it became a salad in one of the ingredients. When we then move to the other study, where we looked at heat-treated plant foods, again we look at how often and how many. We can see that 77 of the samples in that study was vegetable products, and 53 was cereal products. And this graph shows the average prevalence with a 95% confidence interval around it. And it tells us that around 12-13% of the heat-treated foods have Bacillus cereus in a concentration higher than 10 CFU per gram immediately after heat treatment. So we can also have a closer look into, for example, the cereals. And here we can see that rice has a lower prevalence than pasta, again has a lower prevalence than couscous. And This indicates that we might need to use different strategies for these different foods to be sure that we know that the product is safe. But so far, we have used it as a group of cereals in our evaluations. So now, when we talk about how many bacteria in a food, we often try to estimate a distribution. And for bacteria, we use what we call a log normal distribution. And this is an example of how it looks when we show it as a cumulative probability distribution for the vegetables and for the cereals. So the way actually to think about these graphs is the further to the right, you have a higher concentration, and the steeper the slope or the middle of the graph is, the lower the variation will be in the data set. So to give you some examples for these two organisms, we can read here from the graph that for one CFU, to have more than one CFU of a Bacillus per gram, 90% of the cereals has that and 70% of the vegetables has that. But in the other end of the scale, when we have higher concentrations, more than 100 Bacillus cereus per gram, we can actually see that the distribution shifts. And here it is the vegetables that has 10 times more often higher concentrations. So, and this having this knowledge can help us in the end to establish a safe shelf life, for example, for products like this. So this was the results I wanted to share about the how often and how many. Now I will move on to the growth part of our studies in our research group. And first, again, I will like to show you what to expect from the literature, and then show you what to expect from a model that we have developed in our research group to about the growth and non-growth of Bacillus cereus. So usually we tend to use guidelines when we want to produce safe foods, and we have this example for Bacillus cereus that tells us for 3 environmental factors: the temperature, the pH, and the salt content in the food product. What is the inhibitory limit? So when will Bacillus cereus stop to grow? And if we have everything else optimal in a food, and the temperature should then be below 4 degrees, then we can stop the growth of Bacillus. When it comes to pH and everything else is optimal, the pH value should be 4.6 or lower to stop the growth, and for salt it has to be more than 10%. So the reason why I say this optimal several times is actually, if you think about it, our foods, they are not stored at optimal conditions. They are actually stored at suboptimal temperatures for growth of bacteria. And therefore, we at DTU have developed a model that can tell us the combined effect of having low temperature, different pHs, and various salt concentrations. So, and I will show you an example of that. So this is how it can look like, the output from the model, where we at the x-axis has the storage temperature of the food, and on the y-axis has the product pH. And then all the combinations that fall within the red part of this graph is where we have growth of Bacillus cereus when we don't use salt. And just to pick one example, we could look at 5 degrees, storage at 5 degrees, and then we need a pH value of 5.1 or less to be sure that Bacillus cereus will not grow in the food. So now this is higher than the 4.6 that the guidelines say, but it's still a relatively low value when we talk about foods. So you might now say, what if we add some salt? So I have added some salt, and I have chosen to show you the example of 3.5% salt in the water phase because that is what we use for other pathogens. That is what we use for Clostridium botulinum to stop that one from growing. So as you can see, of course, the pH value increases. It increases to 5.2, so that's not very much. And this tells us that it will be very difficult for us to design foods only based on pH and salt and low temperature where Bacillus cereus will stop to grow. So we need to find an alternative approach actually to find a way to produce safe plant-based foods. And what we can do is we can try to establish what growth of Bacillus cereus is acceptable. Because if you think back of the level that EFSA said would cause outbreaks, it's a relatively high level. So there is room for some growth of this bacteria in the foods. We just have to estimate how much. And we do that. This is an example of an approach we have used. We do that by knowing the distribution of the bacteria from the beginning of the shelf life and knowing when it is critical. Then we can use simulation methods, and we can estimate how much acceptable growth we can allow. The idea with using simulation methods is actually that we need to define an acceptable probability to do that. We need to have some kind of cutoff value where we think this is safe enough. And this example that I will show you is an example where we said that we will have less than one product out of 100,000 products exceeding 100,000 Bacillus cereus per gram. So this is one way of expressing this. And when we use this together with the data we have for vegetables and cereals, we find that for vegetables we can have 1.7 log increase of this organism, and the food will still be safe. And for cereals, we can have 2.3 log increases. So now, one thing that I forgot to tell you about this distribution that we use is actually that when we applied it, we also get an estimate of the true prevalence. So now I have talked about 40% in the literature, 30% in the salads, and 12% in the heat-treated products. But essentially, this modeling we did here told us that we should expect 100% prevalence of Bacillus cereus in low concentrations. That is integrated into this value where we have estimated what growth could be acceptable. So as this is a relative number, now we don't have to worry so much about the numbers of Bacillus cereus. We can worry about how is the food characterized, because that could help us design a food where growth can be controlled. So we need the models to do that. So now to sum up, what is actually the role of Bacillus cereus in plant-based foods that I have talked about? First, Bacillus cereus will contaminate plant-based food from the soil. And it looks like from the literature data that we should expect around 40% of plant-based foods harboring Bacillus cereus, but the Danish study actually suggests that we should expect all plant-based foods to have Bacillus cereus just in low levels. That means that we need to control the growth to have a safe food. And we do that by defining acceptable growth limits, and we use simulation methods for that. And then we combine it with our validated growth models for estimating of safe shelf lives. And so far in our research group, we have developed a model that is validated for cereals, and we are currently working on expanding the models to be used for other plant-based foods. Thank you for your attention.
Thank you, Tina.